Linda has been working at the CSIRO since 2001 after completing her PhD at Curtin University of Technology in Perth, followed by a short postdoctoral position with the then Department of Conservation and Land Management. Her research is primarily focused on conserving and restoring the long-term prospects of Australia's unique floral biodiversity. Her most recent research is assisting NRMs and NGOs establish good seed sourcing and production practices to help restored populations respond to changing environments. Dr. Broadhurst continues to publish in national and international peer-reviewed journals, in more than 40 peer-reviewed papers and book chapters, as well as numerous reports and plain language information sheets. That's pretty impressive. She's also been an associated editor with Australian Journal of Botany since 2009. That's very impressive, Linda. So the presentation we're going to have from Linda next talks about why we need to think about genetic diversity in restoration, including the use of local provenance and how climate change is likely to change how we restore plant populations now and into the future. Fascinating. Linda, I'll hand over to you.
Thanks, Hannah. So this is going to be a bit of a tag team with me and Hannah. Hannah's going to do the slides for me because the clicker doesn't work. So thanks for coming. I really appreciate your interest in genetic integrity and bush regeneration. I'm hoping by the end of this talk you'll be persuaded that it's one more of the complexities that you might need to deal with when you're trying to do your bush regen, which I know is already challenging. So, no surprises to an audience like this that we've really changed the face of Australian biota, not just the plants, the biota in general. And so those two maps tell us where the biota has mainly been changed. So the top map is the modelled vegetation pre-European arrival, and then the bottom map is a little bit old now, but it's 2005 where we have vegetation left in significant parts. And so you can see all those white areas around the eastern and western seaboards are associated with where pretty much our grain belts and our farming belts are. So that's a bit of a challenge for plants, especially because those white bits represent places where there is no genetic diversity or there is very limited genetic diversity left. That diversity that was there is gone, and getting it back is nigh on impossible. So we're left dealing with what we've got and making the best of that. So that also changes the way plants operate in their environment, because what we've also done is create lots of small isolated populations, and because plants are static, they don't move around, they're mostly reliant on something taking their pollen away or their seed away, or bringing pollen in or bringing seed in. So if we isolate populations, that means birds, for example, and bats possibly don't fly where they used to anymore, because it's just too far to some of these very isolated populations. So that sets up what's called inbreeding in small populations, and this is a real challenge because it impacts on both seed quantity, so the amount of seed that's produced, and also on the quality of the seed that's produced. And so if you're sourcing seed for restoration, you need to think about both of these things. Can you get enough seed to do the restoration you want? And is the quality of that seed good enough for your grandchildren to still see a plant in the ground of the plant that you've planted? And so I'll just go through a couple of examples of why this is important. And so this is an example. These examples are both from down around where I am. So this is around Canberra. And this is some work that was done by Andrew Young a few years ago, and it's a very small herbaceous plant, the button wrinklewort, Ruidosus leptorhynchoides. There is a ruidosus up around Queensland that flows down into New South Wales. It's got very similar issues. So it's a perennial. It lives about 20 years, but like most of the Australian flora, we don't actually know how long a lot of these things live for. We can only guess. It's had severe habitat loss, and so there's populations around Canberra and there's populations down in Victoria. May have occurred in between there, but that's now pretty much cleared and it's not present that we're aware of. It has quite generalist pollinators, so it's not reliant on a specific pollinator to produce seed. So unlike an orchid that is usually associated with quite a specific pollinator, this is not one of those plants. And it's what we call self-incompatible. And so breeding systems in plants usually range from clonal, so they just vegetatively reproduce, or if they're on the sexual scale, they are either self-incompatible all the way through to self-compatible. And this is one of the self-incompatible species. And so we know in small populations, and a small population for this is 200 plants, which in the region I work in, some people are going, well, that's a big population. But in populations of less than 200, we know we get low seed set. We know we don't get much seedlings being produced and growing through to maturity. And so the populations are continually going into decline, and we have to make a major effort to stop them actually going to extinction. And so this self-incompatibility system is a fantastic genetic mechanism to stop inbreeding. That's its prime purpose, is to stop inbreeding. And so it works like this. If you can imagine that big flower is cut in half, and so it's meant to stop self-pollination. And so pollen coming from the anther onto the stigma is not going to germinate and grow down into the ovules and produce seed. And that's entirely what this mechanism should be doing. So in a large population, you can imagine you've got lots, it's like sitting in this room, you've got lots and lots of diversity, and so you should have lots of different mating types. So in a population that's large, you'll have some orange ones, but you'll have lots of other different genotypes as well, which is represented by the different colours of the flowers. And so if you're a different colour, or a different mating type, or a different genotype, you actually do produce seed. And so we'll just click, and again, thanks Hannah. And so, but if you're related, so if you're another orange plant, you don't produce seed. So there's this genetic mechanism that is to do with recognition of when the pollen sits on the top of the stigma, it recognises that as self-pollen or incompatible, and it rejects it. It isn't allowed to grow through to the ovules. Yeah, one more. And so that, in a big population, produces lots and lots of seed. And that's exactly what would have happened pre-European changes to the landscape. But in a small population, most plants, the seed of most plants falls below the mother plant. Some of it will get dispersed, but most of it falls below a mother plant. And so when you clear vegetation and you leave a small patch, there's a high likelihood that a lot of those plants are highly related. So there'll be seed that's fallen from the plants and will have germinated. And so you end up with a population that's basically brothers, sisters, cousins, second cousins. And so that means, as in this diagram, is that you've actually got lots of those orange relatives. So if you go through the same cycle again, you can't self-pollinate. There are occasionally genotypes that you can mate with, but the majority of them are orange. And so what happens there is you actually get very little seed. And so there are some ethical considerations also for taking this seed, in that sometimes a lot of organisms rely on this seed. So if you take it away, you're actually maybe challenging organisms to survive in that small patch, not just the plants themselves. So this is an example of a plant that's on the other side of the spectrum, so it's self-compatible and it's got very similar characteristics, and Andrew also did this early research. So it lives about the same time, it's also had habitat loss, it's got these generalist insect pollinators, but it's self-compatible. And here the small populations range from about 10 to 400. But unlike the daisy, so Rhododosis, we actually see seed set still in the small populations of 10. They still produce a reasonable amount of seed, but we do get poor recruitment and the populations are going into decline. And this is because these can self-pollinate, so they can produce seed. They're not reliant on that mechanism that stops them selfing. And what this diagram shows, I need a stick. That's all right, I'll just run around. So, what this shows is, this is our population size along the axis here, ranging up to 400. And up here is a genetic measure of relatedness. And so if you're up here, you're highly inbred, and if you're down here, you're less inbred. And these dots represent all the populations. And so you can see if you're a small population, you end up in the highly inbred area. And if you're a large population, you're down in the outbred area. And so what this actually produces is, if you're one of those large outbred populations, you produce plants like this. But if you're in the highly inbred population, you're producing plants like this. And clearly they're not going to survive under a challenging restoration situation. They're going to die out pretty early. And it's interesting, when I talk to a lot of the NRMs and NGOs down around my area, they'll often say, oh look, we had a great year, we did all our weed prep, we did our site prep, fantastic seed collection, and it died. And then they'll then tell me, oh, but it could have been this, it could have been that, but very few of them are actually thinking about it might have been that we didn't have good quality seed. And so now they're actually reassessing a lot of their practices to make sure they know they've got good quality seed before they go to the effort and the expense of putting restoration in, because you don't want that to happen clearly in your restoration. And so that can be challenging to do, but a nurseryman down in Victoria has come up with his own way of doing that, and so he's just doing these little experiments to quality assure his seed before he sells it or sells the plants. And so these trays are another pea species, slender bitter pea, and the numbers represent the number of plants in the population that he collected from. And so you can see if you've collected from a population of 100, you get really good growth. 50 is okay, but not as good as 100. And then 10 and 2 is pretty ordinary. And so this is a very simple experiment he now conducts before he actually sells his plants, because he can now tell people, I've quality assured my seed and I know you will get a great result because here's the demonstration that I've done. So it's quite a good and quick for him way of checking his seed quality and producing a better product to market. One of the things I was interested in a couple of years ago was we know that we're getting changes to the seed quality and seed quantity associated with isolation, and we've been doing restoration in my region for at least 30 years. So how well have we done genetically to make sure that we've reinforced the populations we've restored with sufficient diversity that they're going to go through to future generations? You know, we want these plants that we've put in at great expense to be here in 500 years' time. So do they have the genetic mechanisms available to them to actually do that? So we used yellow box as a study species. It's iconic and valuable for all sorts of reasons. It's quite highly distributed but fragmented. In our region, it's been a very important re-veg species, and so my question really was, does the presence of a yellow box restored site mean it's going to persist for that 500-year time frame? Like most things, we don't know much about it, but eucalypts are generally long-lived. We think this one flowers about every 2 years, but that seems to have shifted in the last few years. We're not quite sure what its flowering cycle is anymore. But we do know that isolated trees produce less seed with poorer germination than woodland trees. So that suggests that the isolated trees have some inbreeding going on that's affecting the quality of their seed. So we did a few things in this study, but the bit I'm going to show you now is just looking at genetic diversity. And genetic diversity is important because when you have a very strong selection event, such as a drought, what you want is lots of genetic diversity, because you know you'll lose some trees or plants because they won't be able to cope with the drought. But if you've got sufficient genetic diversity, hopefully there will be something that can cope in a population, and that will come back into production, and you will actually get this increase in diversity over time, over a very, very long time. So it's kind of a numbers game. You're wanting enough genetic diversity for a strong selection event to at least leave something in the ground to go forward from. And so this is what we did. We took 5 re-veg sites and we sampled the trees in the re-veg sites. We also sampled scattered trees. Scattered trees, I don't know about this region, but scattered trees are increasingly under pressure where I am. But they're extremely valuable in the landscape. They provide habitat. They're a great genetic resource in some cases. And they have all these other effects that are really beneficial. So we also did the scattered trees around each re-veg site, and then we also looked at the seed in the restored trees as well. So that gave us a historic generation, a new generation, and a future generation. So the historic generation is the scattered trees, the new generation is the restored trees, and the future generation is the seed that's going forward and will eventually, hopefully, contribute to producing more trees in the system. So these are just two genetic diversity measures that geneticists like me like to use. They're quite simple. The one on the left is something we call allelic richness, and that's just a numbers game about how many copies of, you can think of it like genes, they're not genes, but how many copies there are of the thing we're looking at in the system. And the one on the right is called expected heterozygosity, and that's important because high heterozygosity is strongly linked with population fitness. And that's simply because if you imagine yourself, you've inherited one set of genes from your mother and one set from your father, and if you've inherited the same copy of a gene from your both parents, you're considered homozygous. But if you've got different copies from your parents, you're considered heterozygous. And heterozygosity is good because if you've got a bad gene that you've inherited, having one gene to mask it actually stops it usually being expressed. But if you've got two bad genes, that's when you see genetic diseases being expressed. So this is good, generally. So what this is showing us is, in terms of the scattered trees for both measures, they're significantly higher than what we put in the ground, so our restored populations, and the seed that's being produced by the restored populations. Now this is a bit of a concern for me because scattered trees are expected to, worldwide, leave the system over the next 120 to 150 years. Just through, they're old, they're diseased, they're dropping out of the landscape. Normally they'd be replaced and they're not. Changing farming practices are seeing bigger paddocks and more precision ag. So they are in the road and so they do get dropped out. What this means in terms of the genetics is, unfortunately, that these restoration projects have left yellow box in a genetically poorer situation than what it was just with the scattered trees. Now that's a bit of a challenge because that means there's insufficient diversity. Even though these patches might be big, there is insufficient diversity, I think, to actually make those useful into the future. So, of course, what we need to do now is go back and add genetic diversity to these sites so that we can improve them to be at least as good as the scattered trees, if not better. I mean, they should be better. Sometimes these scattered trees, there were only 5 of them, and they outdid the diversity in the actual restored trees. Oh, done that one. Thanks. So while that might sound a bit of a dismal story, it does reflect how we used to do things 30 years ago before we actually understood how important genetic diversity was. So often in the past, seed was collected from one to a few trees, and they were often recollected year after year because they were people knew them to be good seeders, so they were reliable. But now we've changed that, so we're starting to collect from 10 trees minimum. 30 trees is much better. That's a challenge, sometimes to find 30 trees. The other challenge is it adds to the cost of seed collection significantly. But I think in the long term, we have to aim high and aim for these 30 trees. The project also showed us that scattered trees have high biodiversity value because they are these genetic reservoirs that we're not actually using as well as we should. And so what we should be doing is catching that scattered tree seed and putting it into restoration projects, but not using it as the sole source, because that's exactly what we've done in the past. We need to mix it with other trees, seed from other trees. And then, of course, we need to go back to those 5 sites and add diversity in so that we can actually give them the best opportunity to shift with change. So that's a couple of case studies that we're starting to build pictures around how to improve our restoration practices. But then we're in a time of quite high change, and that can be for all sorts of reasons. Climate is just one of them. We've got massive weed invasion in lots of areas. We've got changing farming practices. We've got increased nutrient load. So it's not just climate change, there's a lot of change happening. But in terms of seed, there's often this assumption that it's plentiful, and anyone that's collected seed will know that's not true. It's actually often quite erratic and environmentally driven and can be hard to collect. It's primarily unavailable for pretty much all of the species that people want to use, which is a real challenge because that leads to people choosing another species, which may or may not be appropriate. And it also means that, especially in the area I work, we're reliant on 20 to 30 species to be revegetating the landscape. And in my mind, that's landscape homogenisation. If we only have 20 or 30 species, where's our redundancy that you would have in a natural system where in a yellow box community there might be 300 species, so that if something drops out, you've actually got something still there. So we're reliant on this 20 to 30 species, which is high risk in my mind. The other challenge is that people assume that seed is of equally high quality, and we've seen a couple of examples of why that might not be the case. And that it's all genetically diverse, but if we're collecting from these inbreeding populations, then we've got a real challenge for the future. And so we reviewed a reasonable amount of literature just to try and come up with some predictions of what we thought was likely to happen over the next sort of 30 to 50 years. And these probably may be more appropriate down south than here, but our prediction is that there's actually going to be less seed to work with. So we're predicting high temperatures and less rainfall in the region I work in. That means high seed abortion, low seed quality, low seed volume. So there's going to be less to work with, so we have to get cleverer. And then, as I've said before, if there is less seed, we have a real ethical dilemma here about can we take that seed because other organisms need that seed as well. So we're trying to improve our environment, but we need to think about other organisms as well. The other thing that we think will happen is change planting times, and this is already happening in the south and in the west, where farmers are already on board, already see seasonal change on a regular basis. So some of the cropping times are shifting, suggests our re-veg times of planting will also change. And we also think there's a chance of increased planting failure because soil temperatures are likely to increase, and a lot of things germinate according to a soil temperature cue. So that will be challenging for them to germinate and grow if the soil temperature is not suitable. There's also supposedly less soil moisture, and so the other change is that we're increasingly going to be impacted by these odd weather events. So, you might get fewer frosts, but they might be deeper. You might get increased rainstorms. You might get increased flooding. So this is just a diagram we developed, and under a ideal system, and this doesn't work for everyone because if you've only got 1 year's money, you can't do 2 years of prep. But in an ideal system, this is something that Bindi Van Zella, when she was at Growing Australia, helped me develop about. In an ideal system, this is how you would prepare and plant your seed. But we think under new conditions, we're actually going to have to do another step at the front and another step at the end because we have to become much more adaptive than we currently are. So the step at the front is to do what farmers do and evaluate what they think their success for the year is going to be. So they've got lots of tools for doing this on their property. They can put in things like price, weather, which paddocks they want to use, whatever, and they can come up with a risk of what it is. Should they put lambs in or should they not? Should they put canola in or should they put more wheat? Whatever. We don't have any tools like that, but there's a few places I'm going to show you where we might get some information. Then we go through the cycle, but at the end, the critical thing that hardly any of us get to do is actually evaluate the success of our projects. So I will say tens of millions of dollars gone into re-veg. Pretty much don't know how any of it's gone, whether much of it has been successful. And I think for us to be adaptive in a new world, we have to actually build that into all of our projects and build it in for the long term. It can't be something that happens at the end of 3 years because a lot of the effects that we may see could be in 10 or 15 or 50 years. So somehow we've got to build longevity into our restoration as well. So if you're trying to figure out how things are going to go in the future, as I said, there's no tools to help us at this point, but there is a gap in the market for anyone that's interested. But The BOM, of course, have some places that we could look, including their climate outlooks. There is an Ag and NRM page that gives you some information, and then there's also things like the rainfall and temperature outlooks. So that's all helpful, but it's not going to predict success. It's just going to help you manage risk into the future. The reason about the underlying mechanism of managing risk, though, is dependent on your funding agency allowing you to manage risk. So if your funding agency is insistent that you get plants in the ground, this is going to be much more challenging. But they are conversations, I think, that are very important to have with funding agencies about, this is a long-term program. Restoration is not a 1 or 3 year cycle. This is a 50 to 100-year cycle, and somehow we have to be managing for that. So, well, like I said, using climate forecasts are not going to guarantee success or failure, because that's clearly based on lots of things. But if you think there's a risk, then you can actually accept the risk and go ahead. You could scale back projects, so you could reduce the number of species, plant fewer sites, or you could reduce the area that you're going to plant. The other thing you could do is just defer the project until conditions improve. And that's the one I think most people find pretty challenging, especially funding agencies who like to see things in the ground. And I can tell you a story of under NHT1 that some of us are old enough to remember. I know an NRM group that actually knew there was a big risk of failure 1 year, and it was back in the days when a tree went in with a milk carton around it. So they planted milk cartons that year, so that it looked like they had done their restoration, but they didn't waste their resources. So there are ways of getting around it, I guess, if you're creative. But of course, to do some of this means that you have to talk with your funding agency. The community has expectations for you to do things as well, so it's important to have the conversations with the community. But we're actually at the moment trying to have a revision of the funding policy that allows people to take these options. And it's a hard road, we're not getting much traction, but I think it's critical in the future that people be allowed to defer a project if there's a high risk of failure. Why would we put taxpayers' dollars into something we know is going to fail? Because we're expected to put taxpayers' dollars into something. So, changing tack now. There are some genetic considerations that a lot of people are concerned about, and one is provenance, and so that's often thought as the geographic scale over which you can move seed. And some of the reasons around that is if you bring seed, so from North Queensland down to here, that it can't survive the current conditions, that's maladaptation. It might be that you bring two genomes together that have not been together for millions of years, and that'll produce some poor offspring. That's called outbreeding depression and is given a lot of traction, and a lot of people think that it's a major concern. A recent meta-analysis around the world suggests that it's not as important as people have thought in the past. There is certainly some evidence for outbreeding depression, but not every species has outbreeding depression. The other risk is hybridisation, so you might bring something in that you think is the same species but may not be, and you might hybridise, and that can have positive and negative effects. The other thing a lot of people don't necessarily think about is that you bring in a superior genotype. So we know in some populations there are genotypes that will do very well wherever they are, and if you introduce one of those you may create a new weed for yourself. And then there are another issue that is less important here but is important in the Northern Hemisphere is inappropriate timing. So you may get something that flowers at a time that is different to when the pollinator is available, and so you won't get pollination. If they don't co-occur, you don't get pollination. And then the other thing that I've been talking about in terms of genetic diversity, we often call it adaptive evolutionary potential, but that's fancy schmancy sciencey mindsy way of saying genetic diversity, and that's what we want to put lots of that into the environment, so that we do build up this broad base of diversity that hopefully can cope with any change that's thrown at it. So provenance, it's not a new concept. It's been going since the 19th century, and it's primarily comes from the forestry literature. So that was a lot of common garden experiments around the globe that foresters did. So they would go and collect seed from across the range and plasticity than we actually probably give them credit for. And then adaptability is not just about climate. That's the thing in our minds at the moment. But those two pictures there are actually adaptation or lack of adaptation for disease and pests. So it's not just about the climate. Other things that can drive local adaptation or not can be soils, altitude, disease, herbivores, a range of things. So while people are worried about where to shift things under climate change, there is this underlying architecture that influences how we might make those decisions. So what's the Australian evidence for local adaptation? Well, to be honest, it's really hard to demonstrate local adaptation. It takes a lot of years and a lot of money and a lot of heartache. And Nola Hancock from Macquarie, some of you may know Nola, her PhD was based around trying to find evidence of local adaptation. So she actually has done one of the greatest studies so far we've had in Australia, where she's actually done multiple species, planted in 2 field sites, and her first paper says little evidence for local superiority, apart from some traits in 2 species. So, and some of those were collected from around Mackay, I think, and brought down, and some were taken up to around Sydney. So, not strong evidence in those species that we have this very tight local adaptation. Her second paper actually also then imposed 2050 climate conditions on 2 of the species to see how they would react under climate change. And again, no evidence that local ones did any better than the ones that were brought in. And actually, what she did find was that there was evidence that the local eucalypt was actually going to be worse off. It suffered more herbivory than the others, provenances that were brought in. So there's the little twist in the tail about how hard it is to predict these things. Melinda Pickup did this, this is Rhododosis, the button wrinklewort that we saw before. She also did a common garden experiment, and she brought populations from less than 1 kilometre to 600 kilometres together and tested them out to see how they would cope. And again, evidence for local adaptation varied by population and by the trait that you measured. So local populations actually did better for seedling survival, but not biomass. So what that means in terms of local provenance is hard to tell. And then the foreign populations actually did better for producing the number of daisy heads. So if you want something that's going to be a really good flower and then go on to future generations, perhaps you choose the foreign populations. But if you want something that's going to survive better at the seedling stage, maybe you choose the local. And these are the rules we're trying to figure out at the moment about how we're going to provide some information to people about how to choose these sorts of provenances under climate change. And then another story of Eucalyptus tricarpa by Liz McLean and others. They looked at morphological and physiological traits, and not just how things grow, how they operate. And they found some evidence for local adaptation, but lots of evidence that plants are plastic. And that's what those graphs, if you can see them down the side, you can see lots of lines and lots of variability, and none of them seem to do exactly the same thing to give us any predictive power about how this species is going to respond. And so their conclusion is that the extent of adaptive and plastic responses varies among provenances and traits. So that's just adding additional complexity to how we think about provenance. So I call this the provenancing black box. How on earth are we going to cope with all of this? How are we going to make decisions? And so Suzanne Probe has actually come up with a summary of the different types of provenancing that could be done. And so climate adjusted is where you look across a gradient and you choose your provenances across that gradient, and then you plant them in your restoration site, which is the star in each of those diagrams. Or you could go completely local and not worry about anything else. You can also do composite, where you choose most of your seed close to your site, but then less seed as you go further away. You can use the admixture, where you choose your site and then you just go in 2 directions and get equal amounts of seed. Or you can go highly predictive and go, that's what my climate is going to look like, I'm going to get seed from there. So that's useful, but still leaves you with making decisions. And I think what we need to be careful here is thinking that different species may need different approaches depending on what they are. And we're in the stage now of trying to figure out what those different approaches might mean for different species. But if you're hoping to do bet hedging, which I think we're in the phase of trying to bet hedge, probably the climate adjusted, the composite or the admixture give you the best bang for your buck. But having said that, I think if you're really interested in doing some of these provenancing types, we're actually setting up landscape experiments at the moment. We're looking for partners who are willing to actually try something and put it in the ground so that we can follow it, or at least help people follow it into the future. So I suggest now, if you're thinking about it, partner with a researcher. So that doesn't have to be me. There are lots of researchers up here that I can put you in touch with. One of the key things to this will be, however, maintaining meticulous records. So we're going to have to know very good details about seed sourcing, germination rates. Again, you might want to partner with a researcher around that. The reason for the good record keeping is that the benefits or the non-benefits of this may not be in, certainly not my lifetime, in 50 years' time. They're going to be someone else's, and so they need to know exactly what happened when we put it in the ground, and somehow making sure that legacy is going to continue. It's very challenging, especially in the NRM sector where there's a lot of turnover of staff. People move jobs quite frequently, so that's a big challenge, and that can be one of the benefits of partnering with a researcher, is that they sometimes have that longer term legacy. The other thing we have to think about is back to seed stockpiling. This was a big deal under NHT1 and NHT2, and then because the seed banks didn't become self-sufficient, they've all pretty much closed. Huge waste of infrastructure, so we have to be careful we don't do that again. But if we are going to have these less regular planting years and we do have to evaluate risk, then we are going to have to stockpile seed so that we can actually plant it when we're ready to plant it. The other benefits of a regional seed bank, which I think were undersold, were that it was a great way to exchange ideas and mechanisms for restoration. People in seed banks are actually highly skilled in what they did, and they could tell you often which provenance would work where, and we've lost that ability. The other thing we can do is seed production areas. So these are putting species under horticulture, and so that means that we know the seed's going to be high quality because we're providing the nutrients and the water on a regular basis or as required. Primarily they're being used at the moment for difficult to collect species, so those small understory species are the ones that you go, yep, I'll come back tomorrow and the seed's all on the ground tomorrow. But they do cost money, and for longer lived species it will take a while before you get a return on your seed. So eucalypts can become productive around 5 years if you push them with some chemicals. So you force them into flowering early. You can do that, but otherwise you may have to wait 8 to 10 years. And then if we are under increasing temperature and less water availability, then the costs of running a seed production area are likely to go up. But they do provide you with a good mechanism for ensuring your hygienic integrity. Plant Conservation website, or you can contact the authors for a hard copy if you want a hard copy. Noel has got most of the copies, but I've got a few down in my office, so feel free to contact us. It is quite long, but it starts off with these steps that you might like to think about. So we're not saying you have to follow this, but it's just like a checkbox of things for you to work your way through when you're thinking about your restoration site and your seed collection and future climate change. So some other useful places that you might go to. There's a couple of reports out that we did primarily for the Murray Basin, because I led a big project down there, but the information in those reports is actually quite general and could be applicable to lots of places. There's also the Adapt NRM website, which will help you through lots of different things about choosing climates. There's a great tool there where you can click on a map and you can say, what's the surrogate of my climate, and it will show you what other places in Australia have the same climate to you, and then you can push it up to 2050 or 2070 and it will change those surrogates to show you new areas for change. And if you're looking for more information, you can go to the Terra Nova website, and that's got a lot of information especially for the Murray Basin reports that I've put up there. You can search for lots of information about adaptation under there. And it will cover lots of things, including agriculture and water resources and whatever. So you need to sift through it or use keywords, but it's actually a great repository of lots of information that you might be looking for. And that's it. Thank you.